Doobie-doobie-doo. Good morning, everybody. All right, give people a few moments to roll on in before we start. Okay, so before we get going on eukaryotic gene expression, does anybody have any questions about the trip operon? Actually, I have a question for everybody. Pardon me. Did we go through these? Oh, hang on. I mean, you can't see what I'm showing you. Did we go through these slides at the end of class on Thursday? I have a feeling that we didn't. Be a good idea to do that too. Anybody remember what we did at the end of class? I don't think so. No, I don't think so either. Thank you, Christina. Okay, so just as a recap, yeah, so it's really always tricky because I teach this in two different classes, but I'm pretty sure we went through it in a different way and didn't go through these. So I'm going to spend the first 10 minutes of this class going through kind of a different way of looking at the attenuation of the trip codon, and hopefully that will, help reinforce essentially what we talked about last Thursday. So just to quickly recap, trip codon, trip R repressor, it's an apo-repressor, right? So it cannot bind DNA, in particular the operator of the trip codon, in the absence of tryptophan, right? So it requires tryptophan, its co-repressor, in order to, essentially change shape and adopt the correct conformation that allows it to bind to DNA. Okay, so this is the two extremes, right? On and off. No tryptophan, it's on. Lots of tryptophan, right? So essentially, if the trip operon is going gangbusters and tryptophan is not being used in translation, then tryptophan will build up to a high enough level to shut the trip operon off completely. Now, in between those two extremes are varying levels of tryptophan, and so the trip codon needs to be kind of like dialed back a little bit when those levels increase, right? So this is essentially kind of molecular homeostasis, right? Homeostasis in kind of terms of blood sugar levels, body temperature, blood osmolarity, things like that. Essentially, we have the same deal going on here. We want to fine tune or the bacterium does, the output of the trip codon depending on how much tryptophan is present. And that's done using, let's go back up here, this leader sequence, right? This, essentially, I don't know why it's called an untranslated. It is partially translated, but it's part of the polycistronic mRNA. That's a long word. Don't try saying that when you're tired or drunk. But it doesn't produce a structural protein, right? So this is partially translated, but its ability to be translated is the sensor for tryptophan levels, right? So it's this bit here, and if we look at it in larger detail, right, so you can see the start of the TRPE structural gene over here. And over on the left-hand side will be the operator and the promoter, right, where the RNA polymerase binds, right? So this is transcribed. Now, you remember a while back we were talking about how RNA can adopt secondary structures, right? So it can pair with itself to produce loops and, all kinds of funky stuff. And essentially the leader sequence has 4 regions. And those 4 regions can adopt 2 different conformations, depending on which ones are available to bind. That's the key, right? In fact, that's really like the root of all of this. So essentially, 1 and 2 and 3 and 4 can form a loop, right, which you see here. So we have a hairpin loop with 1 and 2, hairpin loop with 3 and 4. But if region 1 is not available when region 3 is transcribed, 2 and 3 combine together, right? And these 2 conformations are mutually exclusive. If 2 is bound with 3, then you do not get 3 and 4 binding. Right? So if we have a hairpin loop of 2 and 3, you will not get a 3-4 hairpin loop. Right? If 2 is covered up or bound to 1, right? So essentially it's not available. That's the key point. 3 is free, it's kind of hanging in the breeze until 4 is transcribed, and 3 and 4 then form a hairpin loop. Does that make sense?
Yes.
Yeah, so it's really all about whether or not 1 is available to bind with 2. If 1 is not, 2 and 3 bind. If 1 is, either 1 and 2 bind or 2 is prevented from binding with 3. It doesn't actually really matter either way, right? The upside is that the 2-3 loop cannot form. And because these are transcribed in sequence, right? 4 is not transcribed until after 2 and 3 are already available, potentially, right? So 2 and 3 are transcribed first. And so if they can base pair first, before 4 is even transcribed, then 3 and 4 will never form a loop. And so the 2 key things here are, 1, the series of this poly-U sequence, right, which, as you know, pauses RNA polymerase. And it is just after region 4. So if 3 and 4 form a hairpin loop, this acts as a terminator loop, essentially intrinsic termination, right? The same as what we learned a while ago, right, in the last section. So the 3-4 loop and this series of U's after it are a terminator loop. So that's one key point, right? So if this terminator loop forms, transcription is terminated. If it does not form, transcription proceeds into the TRPE gene and the rest of the trip codon and the trip operon, gets expressed. The second key point is the presence of these trip codons here. So these are essentially the sensors. These are the things that allow the bacterium to detect, or the regulatory machinery, because this isn't obviously a conscious effort, how much tryptophan is present. Because if tryptophan is present, it will be added to tryptophan tRNA, tRNA, and that tRNA will be available for translation. So essentially, we're sensing directly the amount of tryptophan present, because that tryptophan will be added to its tRNA and it then will be available for translation. And so if tRNA is present, translation continues past that into region 2, right? So remember that ribosome is a pretty big thing, right? So actually it's probably better described here. Right, so here are those 2, kind of a little bit small, but you can see it very well, the 2 trip codons. And remember, this is RNA polymerase and this is the ribosome. And so these 2 things kind of travel at a similar speed, right? So if there are trip tRNAs available, right? So when this ribosome gets those trip codons, it can incorporate tryptophan into the polypeptide. That ribosome will continue onto, let me just move this out of the way, onto region 2 before the RNA polymerase has finished transcribing region 3. So this 2-3 loop cannot form, right? Because the ribosome is obscuring part of it. So by the time the RNA polymerase has got onto region 4, basically it pauses here, and this 3-4 loop forms and boots it off. So transcription is terminated. This is called attenuation. Now, this is also a probability deal, right? Because at, let's say, intermediate levels of tryptophan, maybe there will be enough trip tRNA around for the ribosome to proceed into region 2. Maybe there won't. So it's kind of like a, ooh, this time maybe we'll get transcription. Ooh, this time maybe we won't, right? So you get that kind of in-between level. Now, if there's very little tryptophan at all, right? So this is essentially kind of, let's say, either right after the trip operon has been turned on, or maybe the bacterium is undergoing, a growth spurt, let's say, they found a lot of nice, yummy food to divide on, and they're producing a lot of proteins, right? So tryptophan levels are very low. Essentially, what then happens is the ribosome pauses, right, on region 1. So you can see that here. So ribosome stops here while the region 3 is being transcribed. And so as soon as the RNA polymerase moves off of region 3 to transcribe 4, 2 and 3 now form a hairpin loop. And because 2 and 3 are formed, 3 and 4 do not. And so transcription is not terminated. RNA polymerase basically continues on into the structural genes, and this whole caboodle just stops. Right, the ribosome dissociates, the polypeptide is released and broken down, and those amino acids are reused again. Right, so this is how we manage that kind of intermediate level, right? And you can imagine too, just by chance, maybe you have a low level of tryptophan around. Occasionally, there'll be enough trip tRNA in so that this polypeptide continues to be made, the ribosome doesn't pause, and then you get the terminated loop formed and transcription is terminated. Just by random chance, right? It's very much a probability game. The more tryptophan around, the higher probability that trip tRNA will be able to come in, and this terminated loop, the ribosome moves on to 2 and the terminated loop forms between 3 and 4. Obviously, as tryptophan levels increase, the probability of that occurring increases until it gets to the point where there's so much tryptophan available that you just don't need the trip operon on at all, and trip R binds with tryptophan. But you can also see that once this is transcription is terminated, the total amount of tryptophan structural genes, the ones that make tryptophan around, or proteins, sorry, will dip. Tryptophan levels will drop a little bit. You'll get a little bit more expression. Tryptophan levels go up. It's just a constant kind of homeostasis, basically, making sure you're producing no more than you actually need. It's really a very elegant way of controlling gene expression. Basically, inherent in the control is sensing directly how much of the product is being produced, which I think is pretty cool. Does anybody have any questions about any of that jazz? Because now's a good time to ask. Don't know if I've shown you this yet. Probably not, because I haven't taught with this one. Ta-da! Isn't that the coolest? Look. Stuffed crocheted giraffe. My Christmas present from a couple of years ago. Anyway, okay. Anybody have questions? If not, I'm going to move on to eukaryotic gene expression. Yeah, Tanya, tripoperon is repressible because is normally on and you turn it off with something. And it's a negative autoregulation. So the levels of TRP, as they increase, decrease expression of the tryptophan or the tripoperon. Whereas the lacoperon is inducible because it's normally off, and so you induce it to turn on in the presence of lactose. And again, always think about the context. You only need lacoperon expression when lactose is present. It's completely pointless any other time. But you always need tryptophan to be produced, because you can't be guaranteed of being on a substrate that contains tryptophan. So it's always really important to think about context. What's the bigger picture, and why does this what needs to happen essentially, right?
Okay, but is it always called like repressible negative? And so then the other one, the lack operant, is going to be
It's inducible.
inducible negative.
No, well, so the lacoperon is kind of negatively autoregulated, right? Because as glucose levels increase, expression of the lacoperon decreases. It's just not as direct as the tripoperon is. So I'd be a little bit leery of saying that the lacoperon is negatively autoregulated, simply because I don't know if we're kind of like talking terminology, like if there's a council of people out there that says yes or no, if indirect sensing of a substrate counts as autoregulation. I don't know. So I would always only ever really refer to lacoperon as being inducible, even though glucose levels is an important part of the fine tuning of lacoperon expression. I wouldn't really talk about it in terms of autoregulation, even though that is kind of what's happening. Do you see what I mean?
Okay, yeah, it's just kind of confusing, but the lack operant, it is like it was something like positive something from the first slides.
Just getting back up. Where are we at? Yeah, that one will do. Make sure I'm kind of on the right place.
Oh, positive regulation, right? Because it's always off.
Correct. Yes.
Okay.
But so one kind of tricky thing to remember here, you can have positive regulation by the binding of something, which turns a gene on. You can also have positive regulation through the removal of something, which allows the gene to be turned on. And so it's all about what in the natural default state is bound to the regulatory regions of that gene. In the lacoperon, there's always something bound there. That something is LAC-I. Whereas in a lot, eukaryotic genes, which are generally positively regulated, there is nothing bound there. Only by the binding of something does that gene get turned on. So that's the difference. That's the subtle difference between plain positive regulation and inducible gene expression. So inducible is all about removing something which is preventing, preventing gene expression. Do you see the difference there? It's kind of a subtle one, but it's an important one.
Yes, it's just like I guess like the vocabulary, like new vocabulary, and like the whole mechanism
Right.
put together. It's complex.
Yeah, it is. It is for sure. And again, that's where the bigger context kind of becomes important. And so generally speaking, uh, in bacteria, if you have just an open operator, right, because almost, if not all, control of gene expression in prokaryotes is done through the control of transcription, right? If the operator is open, i.e. there's nothing preventing RNA polymerase from binding and transcribing that operon, you will get expression. Right, so typically bacteria, you have to turn genes off if you don't need expression. Say bacteria that's on glucose and not lactose in its environment. You have to physically turn that lac operon off because you don't need the products because there's no lactose around. In eukaryotes, you have to deliberately turn genes on. Right, so eukaryotes generally are, I would say, positive regulation, right? You have to physically, deliberately add something to that regulatory region to turn genes on. Does that make sense?
Okay, yes.
Yeah. And so if you think about that kind of sense, it makes a little bit more sense that lac operon is inducible, because if there was nothing there, the lac operon would be on, right? But you don't want the lac operon on all the time because it's a conditional deal, right? If lactose is present and glucose is not, then you want the lac operon to be expressed. So you have to put something there to stop it from being expressed when those conditions are not met. Whereas in eukaryotes, if, I don't need to grow any more arms because I already got enough, the genes that are involved in arm development and specification and stuff like that will simply be off, right? Because there's nothing telling those genes to turn on. You don't have to put anything there, at least on the regulatory promoters. Got to be careful about what I say. But you don't have to block those genes from being expressed by binding proteins to their regulatory regions. They're just not going to be expressed. It's only when you want something to be turned on do proteins bind to it, and therefore that's a positive regulation. So even though the lac operon is kind of positively regulated in a sense, because it's kind of told to be turned on, really a better term would be that it's inducible, because its default state would be on if lactose were present. And so lactose has to induce the removal of the thing that's turning it off. So if you kind of try and just think about, okay, what has to happen? What's the purpose of this operon or gene or whatever? Then it makes more sense how it needs to be controlled and how that mechanism would work. Because otherwise, you're right, it's just a bunch of alphabet soup, right? It's hard to kind of pick out what's really going on. All cool?
Yes.
Awesome. Okay, so let's move on to Crazy Town. All right, so essentially, bacteria have really very careful ways of controlling gene expression. One is that they group all the genes needed for one particular task together into an operon. And so expression of all of those can be controlled in one way, one mechanism, or one kind of overall mechanism. And also they're very careful, very, very, very careful about controlling the level of expression through the control of transcription, right? And so the lac operon and the trip operon give you two kind of very different views on how that can be done in prokaryotes. Now, eukaryotes have much more complex ways of controlling gene expression, right? And that's essentially what we're going to spend at least a couple of lectures going through, because there's a lot of stuff going on here. Okay, so just to do some kind of just basic recap, because I did this with the other class and I think it was helpful. I'm sorry, got up early today to ride into work and be taking pictures of butterflies later, which will be cool. What are the steps between DNA and functional protein? So if you cast your mind back to that kind of flow chart that I drew up on my mini whiteboard, what are all the different steps? No particular order, just shout out some as they occur to you. What things, need to happen in order to get a functional protein at the end from a gene in DNA? Make you think. Doesn't matter which order they're in, just shout out whatever ideas you have.
So it's from DNA. They go through mRNA, and then it could be like protein or not
Okay.
functional protein.
So let's go back to one step at a time. Okay, so what's needed to get pre-mRNA? Okay, so we've got control of transcription from Pedro. We have mRNA stability. So if that mRNA is degraded, then you're not going to get translation occurring. You're not going to get protein from it. What else in eukaryotes has to happen to get a functional mRNA, a mature mRNA?
Translation control?
Nope. Well, that's another thing. We'll come back to that. We need mature mRNA. So
control like the RNA processing?
Yeah, exactly. So what steps in RNA processing are there? Yeah, there's 3 of them. One that happens as soon as the 5' end emerges from the RNA polymerase. One that happens once the pre-mRNA is cleaved from the mRNA that's being produced by the RNA polymerase, and one that happens in between. What happens to the 5' end of an mRNA? Nope, hopefully not. Otherwise, it'll get degraded. Going back to your point on mRNA stability. What prevents the 5' end from getting chewed up by RNases?
The cap.
Yeah, exactly, right? So the 5' end has to be capped with a methylguanosine thing. Okay, what happens to after the mRNA is cleaved and what gets added to the 3' end? It's a poly something.
The polypeptide.
Nope, that's translation.
Oh.
Sorry.
What was the question again? What
What gets
after it gets cleaved from the 3'?
what gets added to the 3' end of the transcript? What protects the 3' end?
Oh, the poly A tail.
Correct. And the poly A binding proteins. Good stuff. So what happens in between in terms of RNA processing? What has to happen before that mRNA can be translated into protein? What do you have to remove?
The non-coding regions.
Kind of. Which ones in particular? Because there are 3 different types of non-coding regions in an mRNA or pre-mRNA. Very specific word.
The introns, you need to splice them.
Yeah, good stuff, everybody. And you too, Leviat. So you need to splice those introns out, right? And so RNA processing or mRNA processing is an important part of control of gene expression. Actually, it's a very funky part of control of gene expression. And I'll get to that later on when we get to that point. I have some personal experience in that, which is kind of neat. Okay, so we have control of transcription. We have control of RNA processing. And we have control of mRNA stability. I'm sorry, need my second coffee. Oof. Okay, so now we have obviously we also need to export that mRNA out into the cytoplasm from the nucleus. I'm not sure that's a thing that's specifically controlled. I think that just tends to happen once conditions have been met, but there may be some tweaks that can occur. Hello, Tony. I miss using you as a guide as to whether you're still taking notes or not. I'm just kind of guessing, so I don't like talk too much.
Sorry, I was taking a shower, so I was not ready to put my camera on. I was here, but I was like combing my hair and stuff.
You're attending class in the shower? That's impressive.
Oh, but I was getting ready, like to put the camera on.
Gotcha. Well, ask no more about that. So we've got our functional mature mRNA in the cytoplasm. What next? Actually, Tanya, you already said this. What happens next?
Translation control.
Right, translation. Exactly. Control of the initiation of translation. Right, which if you cast your minds back, is controlled by 5' cap binding proteins and by poly A binding proteins and other proteins that and mRNA, actually not mRNAs, other RNAs potentially that might bind to 3' UTR. Okay, so even though control of translation isn't as big of a deal as a control of transcription, it does occur. Okay, so now we have a polypeptide. Is that polypeptide functional?
It may be, but not
Might be.
100% sure.
Right. Quite often not, actually. What else has to happen?
The phosphorylation? No.
Could be phosphorylation, could be other things getting added to it. Kind of sugar residues, like if it's a glycoprotein, on the surface of a cell, things like that. Um, talk about insulin. Give the example of insulin. I can't remember if I did. Biff, where is insulin needed to do its job? Right, let's say if you're a diabetic, where would you inject insulin? Would you kind of inject it into your butt? Or where does insulin need to be to do its job? If it works like a hormone, essentially.
Would it need to go like to, wait, the endocrine system? But
Yeah, it's a diffusible signal, right? So what is insulin getting transported around the body in?
blood.
Blood, right. So, but insulin is produced in the beta cells in your pancreas. So that insulin protein, which is actually cleaved, right? So cleavage is another part of protein processing. But another part is getting that protein to where it needs to be functional. So insulin that stays in beta cells is not functional. Even though it could function, it can't function until it's released into the bloodstream. Right? So where the protein is a really important part of how it can function. The various proteins that COVID-19 uses to get into your cells, particularly mucosal cells, right? Those have to be on the surface of the protein of the cell facing out, right? Otherwise, the virus wouldn't recognize them. They're there to do other things. I'm not entirely sure what other things, to be honest. Some probably antigen recognition or uh, I don't know, actually, to be honest. I should probably look that up. But those proteins have to be in the membrane facing out, right, for them to function. Right. And so if they're inside the cell, they're not going to be doing anything because they're not in a position to do their job. Right. And similarly as well as with mRNA, how long those proteins hang around for is another way of controlling their function. So again, going back to insulin, right? Insulin and glucagon actually is its opposite, are the two key players in blood sugar homeostasis, right? Maintaining your blood sugar within a particular band, right? It just kind of bounces around in that band, right? You eat a lot of food or you chow down a chocolate bar or something and your blood sugar goes up and then it's brought back down through the production of insulin, right? If you're waking up in the morning like me and you're like, oh my God, I'm so hungry, right? Your blood sugar levels probably drops, right? So you produce glucagon so that you turn glycogen into glucose, right? So it's constantly bouncing around within that. So do you want insulin to hang around? Once it's done its job to tell your muscles and your liver to take up glucose, do you want it to just keep kind of floating around doing the same thing? No, you don't, right? You've got to get rid of that insulin because otherwise, you'll overshoot on the bottom end, right? You'll take up too much glucose from your blood and then you'll go, what's that, hypoglycemic, right?
Isn't that because I had like pregnancy diabetes, and my doctor
Uh-huh.
said like not to worry about it, but it was like my body wouldn't sugar, and then it would like kind of like explode the levels of insulin just like once, just like yeah, just for like nowhere going really high, and then that's what was shooting like my blood sugar level, something like that.
Right, because so that means you get kind of like faint and woozy or something like that after eating.
I don't know. I don't know. I got that, and he just told me that my insulin level was like, it was really like the sugar level was low, and then suddenly my body will make like a lot of insulin,
right?
like really like spike amount of insulin. So it kind of sounds like what you're saying. Maybe my
Yeah,
insulin was hanging around.
yeah. Well, I don't actually know why um, pregnancy pre-diabetes happens. I can only imagine that it's something to do with, changing hormone levels and the disruption of having essentially a large foreign body inside your body, right? Because I mean, there's a lot of really funky things that happen in pregnancy that deal with that. Because if you think about it, you know, you're able to detect self, right, and not destroy it. So your immune system doesn't destroy yourself, but your child inside you is half not you, right? Essentially, it's half of your, the partner that contributed to that fertilization of that oocyte. And so your body has to, in some ways, kind of control itself so it doesn't recognize that baby as being a foreign body and attack it. And so there's all kinds of funky stuff that go on during pregnancy that I don't actually fully understand, to be honest. And I think that pregnancy pre-diabetes is part of that. It's just like whatever's knocking your homeostasis mechanisms out of whack, basically. So anyway, so protein stability, right, and turnover is an important part of controlling the levels of functional protein. Right, and so this is something that's these two slides here are really out of it do a really good job of going through. And so essentially, I haven't even talked about this, right? So in many contexts, not all of them, but many cases, the cell receives a signal, right? So it receives some information from outside the cell that tells it to do something. It might be to make more insulin, right? If in case of beta cells, it might be the presence of insulin that tells a muscle cell to take up glucose. Could be lots of different things, right? Could be a signal that's generated internally, right? If we're talking about cyclic AMP, for example. But the very first step is controlling the type of chromatin that exists around where that gene is in the chromosome. And so this is a real high level concept for controlling gene expression, right? It's really big picture stuff. And so essentially the idea here is that if the DNA that contains this gene is highly condensed heterochromatin, it will not be turned on because proteins that are involved in turning it on, control of transcription, cannot get to that DNA. So the very first step has to be to tell that kind of tight knot of heterochromatin to loosen and kind of unwind and make that DNA available for transcription. And although I've told you that eukaryotes don't group genes involved in the same process, pardon me, together in the same way that prokaryotes do, they kind of do in a way, right? And so rather than being like all right bang next to each other and to make a polycistronic mRNA, instead in eukaryotes, those genes tend to be clustered in a same region of a chromosome. Not always, not all of them, but many of them are. So for example, it's so my usual analogy is if you've ever been in those libraries with the roller shelves, I know they're typically in like the more storage rooms or stuff that isn't used very frequently. Essentially, you have a whole bunch of books on a particular subject that's in one of those kind of roller things, right? And so when you need access to that, you open that up, you can get to it. And then once you've done using that information, you shut it again, right? So you can access somewhere else. And that's essentially in a way what happens with eukaryotes, right? You can control entire programs of gene expression. We're talking about multiple genes that are involved in, uh, let's say, eye development, right? Because once you got your eyes, that's it. You're not getting any more, right? You just get two of them. But there are a whole series of genes involved in choreographing eye development. It's an extraordinarily intricate process with multiple steps. Many of the genes that are involved in that are going to be on a similar region of the chromosome, so that once eye development has finished, that whole region can get shut down because you don't need it anymore. Now, this doesn't necessarily apply to all genes or all processes which have multiple genes contributing to them, but it can apply, and it's a very elegant way of shutting down entire, particularly developmental programs. Things that you use once or at one point in development, and you never need to use them again. And then essentially we have the whole series of processes that we just talked about here, ending up with a protein that is where it needs to be and in a form where it can do what it needs to do. Right, and there are lots and lots of different examples that one could use. So the reason, Blair, why we spend so much time talking about this, why this is such a big deal, is essentially this, right? I'm going to blow this up, and because these are what are called confocal micrographs. Get out of it. Sorry, chat box has just taken over my whole screen. Let's stick it over here instead. Right, so I don't know if these pictures are made with fluorescent protein constructs. I think probably not. Actin is a very, very difficult one to do that with, even though there are ones available now. It might be through immunostaining, right? So basically labeling antibodies with fluorophores, which will then recognize these particular proteins. But this is a neuron. This is almost certainly a neuron that's been grown in a Petri dish, right? So in vitro. And neurons are funky, funky cells. They're actually one of my favorite cells. You know, everybody has their favorite organ and cell and stuff like that. You know, neurons are one of my favorite cells. And essentially we have two different proteins which are labeled with different fluorophores, right? We have microtubules or tubulin, right, which is a cytoskeletal protein, creates the part of the structure of the cell. It's also how products from the soma or the cell body of a neuron are transported out to the synaptic junctions or the growth cones, right? So it's a both structural and also like a molecular highway as well. Tubulin is it's also a funky molecule, right? And actin is another cytoskeletal protein. It's actually part what makes actomyosin the muscle fibers that you use to contract your muscles. But it's also used in other stuff as well. And so this actin is actually used for moving the tip of the neurite, which could be a dendrite or an axon, out towards where it needs to go, right? And so essentially it's very amoeboid-like, right? So you have this growth cone and it just kind of blobs out like that, right? And it's held in out by the actin. And then depending on where a signal's coming from, if there's a positive signal coming from over here, that promotes actin stability. There's a negative signal coming from over here that causes that actin to collapse. So that growth cone turns and grows towards the positive signal. It's really cool. So essentially that's how your neurons get connected to where they need to be. They actually migrate as well, which is also super funky. They kind of clamber over each other in the formation of the brain and its different layers of neurons. Right, so this is a very, very specific to being a neuron, right? They both have this funky structure where they have a cell body and these long projections, and they also are fairly mobile cells, at least early on in development, because they have to kind of wander around to where they need to be and also grow their processes to where they need to go. Now you label the same cells and you look at keratinocytes, right, which help make keratin and, the stuff that, well, Tanya could probably do a better job, but, that makes this, right? Exactly. Thank you, Tanya. And your nails and stuff like that, right? Keratin. It's also that they say they put it in shampoo and stuff. I think that's a load of bollocks personally, but obviously I don't have much of a need for shampoo. Anyway, the cells that form that, same labels. These are now cells which almost look like plant cells, right? They are planar, right? So they're almost two-dimensional, right? So they don't have a shape. They're largely flat. And they have this very rigid junction with other keratinocytes around them, right? So they form a sheet of cells with these are called tight junctions, right? They almost look like plant cells. If you didn't know better, you could make a guess that they were plant cells, but they're not. Here's the nucleus. And as you can see, now we have, again, tubulin, but now the actin is just around the very periphery. Now, the reason I'm telling you all of this is because both these cells have the exact same instructions. They have the same DNA. It is exactly the same between neurons, which can grow up to, or in my case, like 3.5 feet long, maybe less if you're shorter, right? And keratinocytes, which form these very tightly formed layers of cells, right? Which is again part of, their role in terms of protecting the stuff underneath, right? Essentially. But these have the same DNA, right? So their differences, and their differences are huge, are down to which genes are turned on in the neurons versus keratinocytes. They're turned on both in their development, right, that specify one cell type over another, because neurons become neurons because they're told to become neurons, right? Same for every other cell type. That's external signal that we were talking about earlier. But also once they differentiate into these different types of cells, what things do they need? Well, if we're a neuron, we need to produce neurotransmitters, right? Depending on the type of neuron. Dopamine, if we're a dopaminergic neuron. Serotonin, serotonergic, that kind of thing. We might need to produce proteins that are involved in production of a hard protective layer if we're keratinocytes. We certainly won't be producing dopamine, right? Because there's no need for it in these types of cells. Right, so at all points during from their specification, when you're like a wee little, blob of cells in your mother, all the way up to being a functional cell in an adult, different genes will be turned on and off, or rather kept off, and some will be turned on and then turned off maybe later on, to create these very different ones with the same set of instructions. And so all of what we've talked about already in terms of the control of gene expression in eukaryotes leads to that point. Essentially, being a neuron is the outcome of all of those different levels of control of description for all of the genes which determine whether or not it becomes a neuron and allows it to function as a neuron once it does. It's essentially that output. Okay, anybody got any quick questions before we move on? Feel free to ask if you wish.
Just this is like stepping back. So why is the mRNA from like the COVID vaccines, like the Pfizer and the Moderna, put into your muscle? Are they trying to get like to the muscle? What are they
So
trying to do with that mRNA?
essentially what that vaccine is a short piece of instruction telling our cells to make a protein. And so we're basically tricking our bodies into making a protein that looks like the one found on the COVID-19 virus. And so what we're doing, so most vaccines until this is why COVID-19 the Pfizer and Moderna ones are so cool, is that before now, basically what you have to do is either get a actual virus and either kill it or pass it through mice, for many, many generations until it's no longer pathogenic. Or kind of more modern vaccines involve taking a bunch of proteins that elicit an immune response against that virus and cloning them into another virus like varicella or something, something that's somewhat innocuous, right? That doesn't hurt you, but can replicate in you, right? And so essentially you're making like almost like a clone or a parody of that virus, where you have a normal virus with a bunch of bits sticking out of it that look like the virus that you want to vaccinate against. So that was typically how vaccines have been made, which is why vaccine development takes years and years and years, right, to do that. And so this is a very, very, very new technology, which really has been on the sidelines. Actually, if you look up the story, what's her name? Not Kirienko. Can't remember the name. Katalin something or other. Can't remember her last name. Look up the actual person that did the work on this. She was in an academic backwater. She was actually denied tenure at her university, right? So she was like I am trying to become a permanent professor, and she wasn't able to get as much funding or any funding to work on this, and so she was not allowed to become an associate professor, right? Isn't that crazy? And essentially what we're doing is, rather than getting a virus to make these proteins that we then react against, we're packaging the instructions to make that virus inside some lipids, so that when that is found by our cells take that mRNA up and then translate it, turn it into that protein that is part of that virus, and then excrete or secrete that protein into our bloodstream. And so basically now I have, because I've managed to get my vaccine a little while ago, and if it had been a little bit closer, I wouldn't be doing this because I couldn't use my left arm for a few days, which was kind of annoying. But I have a whole bunch of those virus proteins, typically the ones that recognize our cell surface proteins to get into the cell. that I have produced in my own cells and secreted into the bloodstream, like insulin, essentially. And those proteins are then recognized by my immune system, which develop antibodies that then recognize the actual virus if I were to get infected with it. So it is probably one of the coolest discoveries in recent times in terms of biology. And it's absolutely groundbreaking too, because if you get another, say that virus undergoes mutation, which viruses do, particularly when you're trying to kill them or get rid of them, right? There's pretty strong selective pressure for those mutations to flourish. Even if that protein changes through random mutation and natural selection, all the scientists have to do is go tappy 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 tap, make a different mRNA, package it into the lipid droplets, and there's your new vaccine. It is really probably one of the coolest things to happen in the last 10 years, biologically speaking.
I got the vaccine this Friday. So I was reading like the sheets they give you. And it's
Uh-huh.
like, oh, I am RNA. It's like, well, we're studying this. It's like they're skipping a step. Like instead of our body doing that process, they're like injecting the process and then our body like continuing. I don't know. It was weird. I felt like a computer. I felt like a computer. Like they're programming me.
Right. That's funny. Well, I mean, essentially, what's happening in a way is that, I mean, viruses produce mRNAs that our body then thinks are our own to make proteins. That's how viruses replicate, right? They basically trick our cells into making more viruses as if they're our own set of instructions. And so really, in a way, what we're doing is we're just doing a little bit of that, but without the virus. Yeah, which is a super cool thing. Okay, so what's up next is actually a fairly large and involved step. And really the reason why this is so large and involved is because it is complex and multifactorial, right? There are lots of different ways in which one can modify DNA to control gene expression. And so what we're really talking about, yeah, is epigenetics. So this is a new word, right? And what epigenetics basically means is changes to your DNA or proteins associated with your DNA that changes the expression of a gene without changing the DNA sequence itself. And these changes can be inherited. So this is, we always talk about, DNA is actually that's the material that you inherit from your parents, and mitochondrial DNA you get from your mother, and genomic DNA you get from your mother and father. Well, that's most of the story, but actually it's not all of the story, because you also inherit a series of epigenetic marks. These are changes that are made to your DNA and to the histones that your DNA is wrapped around that affect whether or not particular genes are expressed and to what level. Now, there's two kind of really neat things to think about here. One are identical twins. I think I've mentioned this before. But identical twins start off being really identical. Like they have exactly the same DNA. They are really identical. Like can't tell them apart if they dress the same and have the same hairstyle and stuff. But over time, as they get into their 20s, 30s, 40s, and 50s, they start looking subtly different. And part of the reason for that is not obviously just their diet or whatever. It's that they accumulate these epigenetic modifications that changes how genes are expressed, even though they have exactly the same DNA sequence. Those identical twins will start becoming a little bit less identical Not so that you'd say, "Oh, you're not that person's brother or sister," but so that you can start telling them apart fairly reliably. And so those epigenetic modifications are accumulated through the interaction of that individual with their environment, like what they eat, if they smoke, do they exercise, are they exposed to certain chemicals or not? Are they in stressful work jobs or not? Stuff like that. No reason why people go grey in some respects. My parents always blamed on me and my brother, which I always thought was kind of funny. Um, and so that's one example. The other one, and this is even funkier, and it especially applies to any women among you that have had daughters. Tanya, do you have boys only? I can't remember.
2 boys, 1 girl.
Okay. So your daughter was an oocyte in you while you were in your mother. So in the second to last
So,
trimester,
so my mom is basically like also her mom.
in a sense, yes.
Okay.
And your daughter, the oocytes that she now contains, which may not make children later on, she made those while she was within you. So the things that you experience, you know, obviously not a terrible genetics class because she's already been born, so you can't blame me for that. But anything that you experience while your daughter is probably in the last trimester, or was in the last trimester of your pregnancy, is going to change the epigenetic marks on her oocytes, which may or may not get passed into her offspring. So essentially, you experienced the conditions that your grandmother were exposed to while you were an oocyte in your mother, while your mother was in your grandmother. Isn't that nuts? And so they've actually been, it's very hard to do these studies, obviously, because we're not allowed to experiment on humans. That's not ethical. Um, but there have been a few studies on, one was on smoking in Sweden. Another very famous one was on famine during the Second World War in Holland, called the Hunger Winter, which obviously means the winter of hunger, right? In Hollandish, um, Dutch, I think actually that is. But they actually found that women that were in utero during that famine, and basically their food availability went down to about 600 calories a day, right? So what you might eat for your breakfast or lunch is what they had for a whole day. And they found that women that were in utero, like in their mothers, while their mothers experienced that famine, their offspring had poor uh, outcomes in terms of heart disease and various other kind of metabolic indicators. Even though, and that actually lasted potentially, and they've obviously this is a longitudinal study, right? You kind of have to look at this over many, many decades. And this was back in 1944, I think. But those effects persist for at least a couple of generations past that. So potentially you're going to have generations born in Holland this century, which have particular traits in terms of their health, which are the result of their great grandmother going through a famine in World War II Holland. Obviously, over time, it dissipates. It essentially gets diluted over time because you only contribute half of your marks to the next generation. But it's pretty funky that for women, at least, this isn't the case for men. You can have 3 generations in one body at the same time. I just think that's absolutely mind-blowing. Anyway, so really what we're talking about are small changes to DNA and proteins, right? So these could be modifications of histone proteins, right? And we'll talk about what type of modifications those can be. We talk about modifications of DNA, right? And this is something called cytosine methylation. And also the activity of chromatin remodeling complexes, right? So these are a bunch of proteins that essentially reshape chromatin, right? How that DNA is wrapped around proteins. And the overall outcome of that is to control how accessible DNA is for transcription, right? And so essentially one way of thinking about this is here's our TATA binding protein site, TATA binding protein binding site. It's a little bit cumbersome. This is our transcriptional activator protein binding site. This whole protein DNA complex is actually shown a little bit kind of expanded to make it a little easier to see. But if the DNA containing those binding regions is tightly bound to these histone complexes, or is buried like right in the middle of a whole bunch of heterochromatin, these are not going to be available for TATA binding protein or the transcription activated proteins to even bind to that DNA. If those proteins can't bind to DNA, RNA polymerase cannot be recruited to that promoter. No transcription. Right? And so typically we think in eukaryotes of the default state of chromatin as being off, right? So genes are typically off unless, and this actually applies in many levels to control of eukaryotic gene expression. But typically we think of chromatin as being in the heterochromatin state, right? Transcriptionally repressed, tightly wound, you know, DNA that is inaccessible for transcription. And so there are lots of ways in which we can do that. And there's not really time to kind of get started on it. But what I would suggest you do is have a read through these other things, because there's, again, a lot more terminology involved, a lot more words, right? More proteins, more processes. Be good to get at least a familiarity with those things so that when we talk about them, it's not so overwhelming what we're talking about. Because again, we're going to be talking about essentially switching between different states, right? Histones that tightly bind DNA versus histones that don't, that loosely bind DNA. DNA that is refractory to proteins binding to it versus DNA that isn't. Chromatins that's scrunched up versus chromatins that's open, right? And there are a lot of proteins that help that transition, right? Again, just like those sliding shelves in libraries, right? Essentially, you have things which move them open and then they move them shut again. It's a constant dynamic process, which is happening all around the genome, all over the nucleus on a, a minute by minute, hour by hour basis, right? Cool. I think that's more than enough for today. Fabulous questions, everybody. Thank you very much. And see you either tomorrow or Thursday. So again, have a read through ahead on epigenetic stuff, and then we'll go through that in more detail on Thursday.
Dr. Kirk, just before
Uh-huh.
for like next lab, because before it was like the genetics Zoom open lab, but next week do we have to go, or is it going to be through Zoom, or because I got lost with like the winter storm? I know the schedule changed, so did
Right.
that change to go
So
campus?
now that kind of open lab is gone. Uh,
Oh,
Now it's back to in-person, regularly, scheduled. So essentially, the next 2 weeks, depending on which section you're in, is going to be a Eugene lab where we look at the sequences of the PCRs that we sent off for sequencing.
okay,
And so
all
that's
right.
technically you can do that on your own, but I wouldn't recommend it because it's tricky. In fact, one of the things that I'm going to do today is open up that program and just refamiliarize myself with how it works because I'm a little scared by it.
Okay, all right. Well, then I'll see you next week. Well, I'll see you in class.
Cool.
All right,
All right.
bye.
Thanks, everybody.